Hi, and welcome to the second episode of Draw My Science, the video podcast where I interview scientists and I make illustration based on their work. I'm Vale, I'm a physicist and a science illustrator, and in this episode I'm interviewing Dr. Haelewaters, a field biologist who studies a very special type of fungi. These fungi are hosted by flies that in their turn are hosted by bats. This is a very fascinating system and I had a lot of fun talking about it with Danny. I almost felt like I was interviewing a real-life biology-related Indiana Jones. We talk about this system, but also about his other research interests, as well as his career path and how to become a successful academic. On the screen, you'll see me drawing, and I'll leave down below all the information to where to find the illustration in full resolution and how to participate if you're a scientist and you want to contribute to this format. I hope you'll enjoy it.
Hi Danny, thanks for being here. Let's start by introducing yourself. Go with your smartest introduction when you meet someone. What do you say?
Okay. Oh boy. So my name is Danny Haelewaters. I come from Belgium and I'm a biologist. I got my PhD in mycology at Harvard University. Well, it was actually in organismic and evolutionary biology, focusing on mycology. I have a main interest in associations in fungi that are parasites of arthropods. So that's my focus, mostly on these fungi. Although I'm also interested in other things such as understudied groups of fungi and understudied habitats and in multi-trophic interactions.
So
go ahead.
You moved here for your PhD, is that correct?
Oh yes, I did. Yes, I got a bachelor degree in veterinary medicine, and then I realized I didn't want to work in a hospital forever, and so I basically wanted to be a vet in a research team, and I had this dream of going to the Africa and study the big five. That suddenly changed really quickly because I got inspired by a professor in Ghent University who studied mycology, and so
let's step back one second. What are the big five?
The big five animals, like the lion, leopard, rhinoceros, elephant, and buffalo.
Okay. So that's something like specific to Africa, and that's why it's called the Big Five? Is that?
Yeah, I believe so. I mean, this was this dream that I never really fully developed. It's just like I always saw myself as a together with this friend of mine that I had in Belgium, studying large mammals in Africa. So, and the most sexy animal of those is the elephant. So I always saw myself working with those. But then, before you can do research as a vet, at least that's the program in Belgium, you have to do a lot of hospital work. And I just didn't see that happening. And so I switched to biology, which seemed closer to me because it would focus more on the research part. And I still wanted to work with animals, but then I got so inspired by this professor of mine who studied mycology that I then switched topics and became a mycologist.
Okay, cool. Can you tell us what a mycologist is? What is that?
Yes.
What is the study and why it was so interesting for you to switch? What did you find appealing?
Well, a mycologist is someone who studies fungi that are their own kingdom of organisms in addition to animals and plants. And something that people don't really know is that they're much closer to animals than to plants. So that's just something interesting. So the fungi are very diverse. You have the regular mushrooms that occur in the forests with a stem and a cap that everybody really knows about. But we also have many things that people don't really see: parasites, microscopic things, the yeasts, food spoilers. Those you see obviously, but people don't really talk about them or think about them in different contexts. And many fungi are just understudied. And as I'm progressing as a researcher, I'm just realizing more and more how little we know about this kingdom, and I think it just makes for interesting research questions and approaches in just trying to find, discover, and describe that undescribed diversity.
Okay, so what are you studying right now? Can you extend on that?
Sure. So my main project right now, and I'm looking at the number of boxes of alone actually that I need to process, are microscopic ectoparasites of bat flies. So bat flies are blood-sucking flies on bats. So they're parasites, and the flies themselves can be infected by these fungi. And it's a system that before I started back in 2016, I believe, nobody really focused on until back in time, and like I'd say 1917, 1931 was the last publication of a species description in this group of fungi. And so since then, there were 2 papers published on development and host associations, and that's it. And so in the neotropics, for example, we knew 3 species, and that's it. So since I took on this project, I looked at more than 8,000 bat flies, described several species, thought about patterns of diversity, both morphological diversity and phylogenetic diversity, and realized there's just more to this system than meets the eye.
Wow! So you measure different subjects all in the same location, or you travel to do this type of research?
No. I work both with entomologists who send me materials, like this massive loan of thousands of bat flies from Ecuador. But I also travel to countries. So I've traveled to Panama often, where I have colleagues and collaborators. I have an affiliation in David, where I also work locally with students and their professors on different questions. But I have also traveled to Honduras to be involved in surveys. So basically, what I do is I go to the field, I capture bats. Those bats are in the field processed, which means they're weighted, their forearm is measured, they're sexed, they're identified based on morphology. Sometimes we take a wing sample if we're unsure about morphology, so we can check our identity based on DNA. And then we also screen them for bat flies. And then so whenever we have the bat flies in hand, we travel to the laboratory, which is mostly the day after. And then we screen the bat flies for presence of these fungi. And then when I find the fungus or different fungi, then I process them, which means DNA extraction, morphological analyses, and then even further processing, host associations, taxonomy, and things like that.
What does it mean, host association? Is there a preference of certain fly to go to certain host? Is that one?
Yes. So often these fungi, they're grouped in the order Labulbiniales after a French entomologist, Labulben, which explains the difficult name. These fungi are usually very host specific. So we do see that there is a trend for a certain species to be associated with one host, and then for another species, may it be a sister species to another host. So yes.
Okay.
But in some cases, that's not 100% true. So it is always important to have a good identification of the fungus, as well as of the host. So, and that's the reason that I work together with entomologists, so we can always put a good name down for the hosts as well.
So here there are three pieces of the puzzle that need to go together and work. So we have the fly on the bat flies, on the bat, and
Yes.
then we have the fungi on the fly. And then you have you and the team of people you work with, you have to reconstruct this puzzle to understand why certain association happen and where and how, I believe. Is that correct?
Yes, that's absolutely true. So I work with mammalogists to deal with the bats. And I go out and mist net for bats myself, so which is a lot of fun. Bats are so cute. But then the next step is the entomologist part, to figure out the bat flies and the associations of the bat flies with their bat host, because those are also very specific. And then there's the associations of the fungus or the fungi with the bat fly. So yes, there is indeed three parts of the puzzle. And then the last part, of course, is putting it all together in an integrated framework and understanding these different multi-trophic interactions.
How is it to work with so many different people? I mean, is it does it make the interaction faster, slower? And do you work always with the same people, or this team of people change depending whether you are in Honduras or in another part of the world?
Right. I love working with just collaborators. That's one thing. I think I have a collaborative mindset. So I enjoy having to work with many different people, and I enjoy working in different fields too, because it always makes me wonder about using things like jargon, field-specific words, right? You shouldn't always use when you're talking to people in different fields. And so I have that. It's a bit of a balance, I feel like, because it's sometimes difficult to write a paper for a certain audience or write a book chapter, which we've just done. We wrote a chapter for a book about bats. And so you can't just dive into your mycology-specific wordings of things, and so you need to think outside of the box, which I enjoy very much. Do I have collaborators depending on where I collect? Yes and no. I do have collaborators that I work with very, like, a lot, mostly on most of my projects, but then there's also collaborators that I collaborate with in certain areas of the world. So in Honduras, for example, I have joined a team, Operation Wallacea, that was already in place. And so all the people that are working there are just new people that I work with and that I collaborate with. So I collaborate with them to help them with their bat surveys. They're doing these surveys for several years in a row to understand how communities are changing over time. And so I joined their team, but at the same time, they're collaborating with me to help me with my data on the bat flies and the fungi. So it works two ways, if that makes sense.
That's great. What would you say is the funniest part of your job?
The funniest?
Yeah, of this research. What is it? What is that the part you enjoy the most? Is that going on the field hunting, or is that going back to the lab, or is looking at the data? What is the part that you?
I think I enjoy fieldwork the most. I think I consider myself more a field biologist than anything else, just because it's outside of the laboratory. So when I started this bat fly associated fungi project, I started working with collections, which is cool because you have a lot of bat flies without you having to get outside of the lab, right? So it's you could be lazy in a way because the data are there. You just have to sit at a microscope, look at these bat flies and find the fungi and then deal with the fungi. But then on the other hand, you really don't understand the ecosystem that they occur in. You don't really know where they're from or where they were collected or how the associations form, because you don't. Well, that's the way that I feel about it. And so since I've been in the field myself, I just understand this system much better because I see those bats interacting with each other. I see which bats interact with which species, where those bats occur. Certain bats, they occur only in tunnels, while other bats occur just like everywhere in the forest. There's certain bats that you can't capture with low mist nets, but that you have to capture with these high mist nets. And so all these things you only learn when you're actually in the field, when you're doing it yourself. And so that's just what I find so rewarding about doing field work. Also, with doing field work, you have to interact with many people, both locals and collaborators, as well as students. And I enjoy that a lot, to be dealing with those different people.
That's great.
Field work.
So would you say that the field work is also inspiring you on the more brainy stuff, because you get a further understanding and you get new inspiration because now you see an interaction that you would not have imagined or something?
Mm-hmm.
Okay, I have the last question regarding your research, which is typically the difficult one, is that why people should care about this?
Oh, that's a great question. Well, there are two components, I think, to this research. The first one is trying to understand how much diversity is out there. It's just like this really big overarching question. How many fungi are really out there? That's mostly like what lies at the forefront of everything I do, really. And it's quite important, right? Because how can one think about conservation or about protecting landscapes or forests if you don't know what occurs there? Does that make sense?
Yeah, it does.
And then the second thing is these things are parasites. And so I'm always thinking about, well, presumed parasites, I should say, because we don't really know the interactions yet. But we presume, based on preliminary data from other systems of Labobinielis, that they do have a negative effect or some sort of negative effect on their host, on the fitness of their host. And so the idea of studying parasites on bats, and that's the two levels of parasites, the bat fly parasites and then the parasites on the bat flies, is to try to test this hypothesis of the dilution effect, which stipulates that a healthy ecosystem reduces the risk of diseases in a certain area. And so I'd like to test that on these different levels. And that's quite important because, bats provide so many different services. For example, they're important pollinators. And so it's quite important to just look at bat assemblages and bat associates, including these parasites, to look at hypotheses like this one. If that makes sense.
Is this dilution effect something that is specific to this system, or is an hypothesis that is more general to the animal kingdom?
It's a general hypothesis that I would like to make in this system. Yeah.
So it's something that it will help us understand ourselves in a way. Also, in the long term, we are maybe much more complex system since we change the way we live, but in a way it will be okay. That's fascinating. I didn't know anything about this dilution principle.
Yeah, it's a very interesting hypothesis, and the thing is with a system like this. Oh, I haven't told you that the prevalences of these fungi on bat flies are very, very low. We're talking about 2 to 6%.
Okay.
So we have to basically screen 100 bat flies before you'll find 2 to 6 of those infected with fungi. And so it's really hard to get high amounts of data. Well, it's easy to collect a lot of bat flies, but it's not easy to collect a lot of bat flies that are infected with fungi. And if you want to test a hypothesis like that, you need to have, a big enough data set with big enough numbers of bat flies that are both infected and uninfected. So I am working on building this massive data set with thousands of bat flies and to be able to start processing and statistically analyzing so we can test hypotheses like this one. So currently we have 8,000 curated bat flies, which means that we have all the data in place for them. The bat flies are identified, the fungi are identified, and all of their associated data and bat data are there in the database. And we probably have around 5,000 to 6,000 other bat flies that are in different stages of processing. So we have screened them, but we haven't identified the fungi yet. And so we need to put those bat flies all together in a database. I'd like to get at 20 or 25,000, and then we can start doing some analyses.
Wow.
So yes, it's a big undertaking, but I enjoy it.
But then there's a lot of space for data scientists, because with that amount of data, you have to be screening, looking for a pattern that you don't immediately recognize, and it's gonna be a lot of work. That gives me a really good segue for my next question. What's next? Mm-hmm. How long do you think you're gonna work on this system, and do you have a big dream for the future?
Yes. How long I'm going to work in a system? Well, I started working on this in 2016-17, published a number of papers, but I think a lot is still unknown. The thing about studying understudied system like this one is the more you dive into it, the more questions pop up. So you start with one question, but then you're trying to resolve it. And as you're working on getting towards the answer, you're not even there, but there's like 17 new questions that pop up in your head. Like, oh, how does this happen? Oh, how does these associations evolve? Because it's so weird. And so I think I'll be working on this for quite a while. But at the same time, I like to diverge. And so I have taken an interest in other understudied groups of fungi. And so, for example, I work on the Leosiomycetes, which is a class of fungi that are mostly cup fungi. So they're mostly tiny little cups, but they also have plant parasites, bat parasites, actually. One of the fungi that wipes out populations in North America, Pseudogymnascus, is in the Leosiomycetes. And so not many people work in this group. There's also a problem of undescribed diversity. Wherever we go, we find new species. So I did a preliminary fungal survey in Honduras as I was there to capture bats. At night, during the day, I collected fungi. And so almost all of the fungi I found in the Leosiomycetes are new species, which really points me to like, whoa, there's just so much to do here. You
wouldn't expect that after the big exploration time, we're still finding new species.
We've described fungi for about 300 years, I'd say. Yeah, a little less than 300 years. And we're at 135,000 species that are described, which seems mind-boggling, right? That amount. But the number of estimated species is somewhere between 2 and 3 or even 6 million species.
Wow.
So
there is a lot of room.
There's a lot out there that isn't known yet. And so one of these most understudied groups are both the Labylbiniomycetes, the ones that I studied that are associated with arthropods. So, for example, the Batfly project falls in it, but also those other ones that I study now, the Leosiomycetes. And I also, in addition to that, focus on other groups that are understudied or areas that are understudied. So I just applied for a project to study fungi in Siberia, which is a really interesting project. So I like to focus both on groups that are understudied as well as places and localities, and geographical areas that are understudied.
I think it's a very noble goal to study this forgotten species or forgotten environment. I think it's really important to go there. But does it make it more difficult for you to apply for funding? What's the outside approach of the, how is it perceived? Because I'm also a scientist, so I know that there are certain topics that are sexier than other, and they get more funding, and how do you cope with that? Is that a challenge you had?
I try to find applications in the groups that I study and try to find sexy approaches to study some of these groups. And so, for example, in those Labylbiniales that are parasites of arthropods, mostly insects, I have tried to get funding, sometimes successfully, sometimes not, using a biocontrol approach. So one of the most found fungi in that group is a parasite of an invasive species. So it's a species of ladybirds that has been introduced in North America and Europe to as a biological control against aphids that were destroying our rose gardens. But now it has become a pest species itself.
Okay.
So now people are looking for a biocontrol against a biocontrol, right? And so I developed a project in which I use those fungi to see if we can actually see them as biocontrol agents. And with some success, I'd say we're about to publish a paper on that. But that sort of funding that I get for that project also allows me to study then the questions that I'm also interested in with regards to diversity, taxonomy, and things like that. But another thing is you can also study understudied groups or understudied habitats. For example, the lettuce phyloplane, the lettuce leaf surface is an area that seems very common, right, and logical to study, but we haven't really studied it for fungal inhabitants.
Really?
And that's just no. And so here at Purdue, I'm involved in this postdoc project that I'm doing to study specifically that. And we actually found that one of the most common species on the leaf surface of romaine lettuce, which is one of the most commonly
produced and consumed.
Produced products here in the States. One of the most common species of fungi is an undescribed species. We don't know what it does. We don't know its ecology. We don't know its physiology. It's so weird. And so, yeah, I do see that too. So we can actually, apply for grants that then focus on agricultural side of things, even if we're looking at biodiversity. So I also always try to find some sort of applicability, even though there are these days grants from the National Science Foundation that focus on diversity itself, and that's just great. It seems like even though it's hard to make people realize that taxonomy and thinking about diversity is hard, that there's also a movement to emphasizes those fields of research.
It is fantastic. This is really a lot of insight that I'm learning from you today. Yeah, I guess the only thing is left is if you want to share any challenge you had in your career or anything that you learned that you feel like it's an important lesson that you want to share.
Okay, boy. Yeah, let me think about it for a second. I think one of the things I've learned in my academic path, as well as seeing how other people go through their paths, is I think not a single path is the same, and I feel I see these young people, undergraduates, like trying to mirror them to like these big names in research, and they're like, oh my gosh, I haven't done this or that yet, or I haven't published a paper, or my grades are too low, or whatever. And I'm like, you know what? I had low grades when I was in college, and I ended up with a PhD from Harvard. It's all on how you approach things, how passionate you are. Passion is so important, and about craftsmanship in a way. I think being in academia or successful as an academic is hard work. I think that in the first place, hard work, a little bit of luck every now and then, especially hard work, and being outgoing. Not necessarily that you have to go to every party and talk to a lot of people, but have collaborations and be in touch with several people. That at least has helped me a lot, to be in touch with so many people. And not every contact will lead to an actual collaboration, but some contacts, even though we're not collaborators, have instigated other collaborations. And so that would be maybe my guidance, if that makes sense.
Yeah, it does. And I really like coming from you because you mentioned at the start that you switched field early on. So that's already a very non-common path, I would say. Not many people, they go from wanting to become a veterinary and study elephant in Africa to study fungi in Romaine, which is it seems so very different in the spectrum. And I think it's a journey of self-discovery, and you have to be open to adjust your path in a way that best serves you.
Right, continuously, really. And having gone through that program of veterinary medicine, the bachelor program in Belgium, really helped me. It still helps me to be a successful academic, actually, because it was rough. Because it was quite difficult coming from where I came in high school to be on top of all those things. And I think I really learned to learn during that time. And so I think I laid the foundation for all future studies. So for me, it was really good that I did that. I don't think I would have been a successful academic if it weren't for that weird side path. I mean, it looks like a weird side path now, but in my head, it's kind of super logical. I just had to do that side path to be able to become the academic that I have become right now.
Which is going back to what you say at the start. Not every single path will be the same. It's just everyone has his own path, and you just learn from what you have. If you find what you like, then you'll get there eventually by different path that will serve you.
I mentioned that I'm passionate, and I think that's really important. I think as an academic, you need to be passionate about your work because indeed the demands are very high to be successul and to stay in academia. I see many people that I believe are really good academics. I see them, branch out to different fields outside of academia just because it's too much. And I think part of the reason maybe that they're missing this real internal passion about their fields, perhaps.
Okay. How do you balance this being too much with a healthy lifestyle? You have a daughter, you were saying at the start, a wife. Did you manage to find a balance about that?
I try, and it's sometimes better than other times. I think that's probably my biggest struggle, I'll say. The healthy balance. I try to be balanced though. I work a lot during the week, and I have 2 dedicated days that I just work, nothing else. And then the other days I try to make more time for family business, cooking and things like that. And in the weekend I try to be home as much as I can, as much as not going to conferences or invited talks and things like that. Allow that, or field work for that matter. But yeah, having a daughter changes everything, I feel like, or having a child, just because there's so much that I do not want to miss in her development. And so it just, it has changed a little bit though. So yeah, but I try to give my passion to her as well.
That's great.
She goes to the forest and finds fungi.
That's adorable. Okay, I took 30 minutes of your time. You have several thousand of flights to study, ready to be analyzed, so I don't want to steal more of your time. Let me close with tell everyone where people can find you, where they can find if you have a website about this project, or if you have your social tag or anything you want to close it with.
I have a website. It's DannyHaelewaters.com, and I'm active on Twitter. And that's how do you call that symbol? At D Haelewa, HAELEWA.
Thank you again. It was a fantastic chat.
Thank you. Thank you for having me.
I hope you had fun with this interview. Leave your comment and suggestion on how I can make this type of format better. I'll see you in the next episode of Draw My Science. Bye!
You can go to the jungle and describe a new species that nobody will ever get a grip on, right? Well, this thing, they can actually buy the lettuce and have the fungus in their hand, because on every freaking lettuce that we saw, that we analyzed, that fungus is there. So it's weird.